morning, good afternoon, good evening. Welcome back to Chapter 5. We're talking about transport. We finished up talking about passive transport, which doesn't require energy, and active transport, which does. This graphic right here summarizes the methods of transport we've talked about up until now. These are both passive. They don't require the cell to spend any energy. Simple diffusion stuff goes straight through the cell membrane. Facilitated diffusion, the only things that can do that are small nonpolar particles. Everything else that uses, the other things use facilitated diffusion, and this is where they're big and they're polar, so they have to have a channel to go through. Osmosis is specifically talking about the movement of water and not just dissolved stuff. We're saying there's more water out here than there is inside, so it moves from where there's more to where there's not as much. We're still talking about the movement from a lot to not much. This time, these are particles that are moving. This time, it's specifically water that moves. And our last method of transport was active, or our most recent method, and that's where you move it against the gradient. There's not much here. There's a lot out there, and I'm forcing it from where there's not much to where there's a lot. If I'm going to do that, I have to move them against the gradient, and to move stuff against the gradient, you have to spend energy to do so. There are three methods of transport. But if the first method doesn't require the cell to spend energy, and the second method does, well, then what can the third be? The third is called vesicular transport. And the stuff that we were just talking about, passive and active, that's like the difference between paper or plastic in the grocery store. Two different methods of moving stuff, but small, manageable things. Vesicular transport is, do you want to use a pickup to move it, or do you want to use a dump truck to move it? We're not talking about the movement of little things now. Now we're talking about the movement of big things. Vesicular transport is specifically transport using vesicles. We've talked about vesicles before. A vesicle is a pocket of membrane, a little balloon of cell membrane. And these balloons of cell membrane are used to move things sometimes into the cell and sometimes out of the cell. And there are two methods of vesicular transport, whether you're moving stuff in or whether you're moving it out. And our first one is called endocytosis. What does endo mean? Endo means inside. What does cyte mean? Cyte means cell. And osis just means a process. And so a lot of times when we look at words like this, inside cell process, that doesn't make any sense. But you got to kind of rearrange it and use a little creative license here. And it's the process of moving things inside the cell. Process of bringing things inside the cell. And there's a couple different methods of endocytosis, and our first one is phagocytosis. What does phag or phage mean? And phag or phage means to eat. So phagocytosis is the process of cell eating. The process of cell eating. Please be careful. This is not what your cells do when they're hungry. What your cells do when they're hungry is they do that process we talked about before, about how sodium brings sugar inside, using active transport to set up gradients so that the sodium wants to bring something else back with it. That's what your cells use, and that's not what we're talking about. This phagocytosis, what we're talking about in your body, is how your immune cells, your white blood cells, defend you against infection. They see something that should be gobbled up, they see something that's an invader that shouldn't be there, and they will eat it. They eat it to destroy it, eating foreign particles. What the cell will do is it will push up what are called pseudopods up around. They're just extensions of the cell membrane. These extensions of cell membrane will just kind of grow up around the food particle, whatever it is. It could be a bacterium, it could be a virus, it could be something that needs to be destroyed. And it brings it inside a balloon of cell membrane, and then it will gobble up. It'll attach those lysosome things. It'll attach the lysosomes to it and then destroy whatever's inside the pocket. If phagocytosis is the process of cell eating, pino means to drink. This is the process of cell drinking. And there's a big difference between phagocytosis and pinocytosis. Phagocytosis is for bringing large particles inside the cell. Pinocytosis is for bringing samples of fluid from outside the cell. We're not 100% sure why cells do penocytosis. We know they do it. We're just not real sure why they do it. But let's look at a picture of it, and it'll make more sense that way. So what we're looking at here is we're looking at the cell membrane of a really big cell. The top one up here is phagocytosis, and this is that cell membrane, right? And like I said, extensions of the cell membrane will grow up. They basically kind of ooze up like a blob, and they'll flow up and around and surround whatever this food particle is. And notice how food is in quotes. It's in quotes because, again, it's not eating this thing to gobble it up. It's eating this thing to destroy it. And these pseudopods, once they ooze up, and once they hit together at the top, they fuse together, and they make this little balloon. It's called a vesicle. They bring this little balloon containing whatever that thing is now inside. And then what they do is they will attach, like we said, attach lysosomes to it to destroy it. Notice the fundamental difference between phagocytosis and penocytosis. Phagocytosis, the vesicle, is made by extending the cell membrane out of the surface of the cell. Penocytosis is by dipping it inwards. Same basic process once they touch at the top. Once they touch at the top, it fuses together, and now I have my little pocket. I have my little vesicle. The other difference is this is for bringing in large particles. This is only for bringing in samples of fluid. Again, we're not 100% sure why cells will do this, but they will do it. This last one we're not really going to talk about so much. This is called receptor-mediated endocytosis. Interesting little tidbit, not test material. Interesting little tidbit. This is the method that COVID-19 uses to get inside your cell. It attaches to receptors on the outside of the cell, and when they do, that receptor tells the cell, bring me inside. And so it brings the virus inside your cells. But again, don't worry about that. That's not on the test. There are two varieties of vesicular transport, moving stuff into the cell or pushing stuff out of the cell. And our last variety of transport is called exocytosis, and this is releasing things outside the cell. And let's look at that bottom picture because this is demonstrating exocytosis. So this little vesicle right here was just released from the Golgi apparatus. It contains some molecules that this cell was made to release outside, and we said that. We said that anything that goes to the Golgi needs to be packaged to be released outside. So here's my thing that's been packaged to be released outside. This vesicle will bud off of the Golgi and will float and drift to the surface of the cell. And when this little balloon, this little pocket of cell membrane attaches to the surface of the cell, it fuses with it. And now that it fuses with it, it opens up and releases its contents outside. And the fabric of the balloon now becomes a new patch of cell membrane. We're not exactly sure why cells will do endocytosis, specifically rather penocytosis. We're not exactly sure why cells will do penocytosis, but if you look at it, it's just the reverse of exocytosis. Here, it takes a sample of something from the outside, brings it inside. Once it fuses at the top, it now pops inside. You've got a sample of whatever the stuff is outside. This is exocytosis, releasing stuff out. This is penocytosis, bringing stuff in. You notice how they're just simple reverses of one another. This is vesicular transport.